So, hello everyone who already joined our masterclass today, and hello to everyone who's gonna watch the recording of this masterclass. My name is Marina Lova. I'm a part of the service team here at Cube Biotech, responsible for the project management of the service projects. And today I'm honored to host today's Intelligent Infusions masterclasses here from Cube Biotech, created for the researchers in order to give them opportunity and a platform to share and to promote their research with a worldwide audience. Today's speaker is the Renee Chow, and she's gonna tell us about the role of the mechanical forces in the heart valve development. And before I introduce our speaker in a more detail, I wanna show you that if you want to ask the question to the speaker, you can basically type your question in the chat window in your lower right part of the screen. Yeah. So, but please make sure that you change to the question mode from the chat mode so that we can show your question afterwards on the screen. So, as I said, our today's speakers is Renee Chow. I want to give you a bit detailed introduction to her work. So basically, she has done her PhD at the University of Cambridge, where she studied the retinal development under supervision of Professor Bill Harris. And then she started to work as a postdoctoral researcher in the Institute for Genetics, Molecular and Cell Biology in the University of Strasbourg under mentorship of Dr. Julien Vermot, where she started to elucidate the role of the hemodynamic forces caused by blood flow on zebrafish heart valve development. She received also the fellowship from the Lefoulon-Delalande Foundation, and from January 2023, she started as a group leader at the Australian Monash University in the Institute for Regenerative Medicine in Melbourne, Australia. So with this, I want to tell you, please enjoy the talk of Renee. And as I said, if you have questions, you can directly type them in the chat, and please change to the question and answer mode. So please enjoy the talk.
So thank you to Florian and for the organizers of this series for giving me the opportunity to speak today. So I'm a new group leader at the Australian Regenerative Medicine Institute, and I'm a former postdoc of Julien Vermot. And today I'll be mostly talking about the work I did in the Vermont lab, and towards the end, I'll talk a bit about what I'm doing now in my own lab. So I'm interested in the heart valves, which are structures that are essential for unidirectional blood flow. The human heart has 4 heart valves, here, 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 and here. And when they're working properly, they open fully to allow blood to flow through, and then they close completely to prevent blood from flowing backwards. Calcific valve disease is a progressive disease that results in a dysfunction of the heart valve. It most commonly affects the aortic valve, but it can also affect the mitral valve and more rarely the tricuspid valve. And what happens is that abnormal mechanical forces or inflammation can trigger the onset of disease, leading to valve thickening. And when this happens, the heart has to pump harder in order to get blood through a narrow opening. As the disease progresses, you get these calcium deposits and changes in the extracellular matrix, and this leads to valve stiffening. And at this point, the valves no longer close properly, and you get backwards flow of blood in the heart. This leads to further cardiac complications, and unless you replace the valve, this eventually leads to death. There's a lot of clinical incentive to study how the heart valve forms in the embryo. This is because although calcific valve disease is a disease that mostly affects people aged over 75 years of age, it involves the abnormal reactivation of developmental pathways. And also, if you have congenital heart valve defects, this increases the likelihood that you're going to develop the disease. Most of what we know about heart valve development comes from studies in the mouse. And here's a schematic of atrioventricular heart valve development. This here is the myocardial layer, the muscle layer of the heart. This here is the endocardial cell layer, the cells lining the lumen of the heart. And between these two cell layers, you have the cardiac jelly, which is mostly extracellular matrix. What happens is that some of these cells undergo a process called endothelial to mesenchymal transition, or endo-MT. They lose the cell-cell contacts, they lose the apical basal cell polarity, and they migrate into the cardiac jelly, where they proliferate, and then eventually differentiate into the valve interstitial cells. Around this stage, you have a step called delamination, where you get these holes forming in the myocardial layer. And then this part of the tissue splits from the myocardial layer to release the valve leaflets. And these cells here eventually become the papillary muscles. This entire process happens under the influence of blood flow and heartbeat. And during my postdoc, what I was wanting to find out is, what is the role of mechanical forces caused by blood flow in regulating this developmental process? Because of the limitations in the mouse in terms of live imaging, we use the zebrafish as a model. And the zebrafish has a simpler heart. It's only got two chambers, the ventricle and the atrium. And for my work, I'm looking specifically at the superior atrioventricular valve leaflet. To answer the question, what is the role of mechanical forces in heart valve formation, I have three smaller questions. The first is, how does the heart valve form in the zebrafish? The second is, do forces play a role in the process? And third, if it does play a role, then what's the mechanotransduction pathway? So a number of previous studies in the zebrafish has come up with this developmental sequence for atrioventricular valve formation. You initially get a single layer of endocardial cells overlying the cardiac jelly, just like you have in the mouse. And then some of these cells undergo collective cell migration into the cardiac jelly to form this folded tissue structure here. And then it is thought that these cells undergo deadhesion, so they lose all their cell-cell contacts, and they eventually differentiate to become the valve interstitial cells. And at the same time, you have changes in cell shape and proliferation that lead to leaflet elongation, transforming this structure into this structure here. And what you will notice is that in this current model, there's no delamination step, so there's no splitting of the cardiac cushion. But when I started doing imaging experiments, I found out that this model here is incomplete, because at this stage where it was thought that the cells lose all their cell-cell contacts, I actually see that there's an increase in the cellular organization. And also the first valve leaflets that I observe are actually 2 cell layers thick, which kind of goes against this idea that all these cells that migrate into the cardiac jelly differentiate to become valve interstitial cells. And you can see that the valve is 2 cell layers thick more clearly in this image here. So this is the same embryo, the same valve, but here I've stopped the heart using the drug. And this is the first layer, which is the outer layer of the valve leaflet. This is the second layer, the inner layer of the valve leaflet. And here's the third layer, which forms the atrioventricular canal wall. What we think is happening is that after the cells migrate into the cardiac jelly, you have this reorganization process, and then this layer of cells splits from this layer of cells to give rise to these 2 cell thick valve leaflets. And we managed to capture this using time-lapse imaging. So this is the same embryo, but every single half an hour, we image the entire cardiac cycle. So at these 2 time points, even though the heart is beating, all the cells remain stuck together. So you have a structure that looks something like this. And from this time point onwards, you start to see a gap labeled by the blue arrow every single time the heart beats. So this layer of cells are separated from this layer of cells, and you have a gap between these 2 layers. And here, the gap is just a little bit bigger. And so what this kind of suggests to us is that this process is very, very fast. It occurs within an hour, even though this event can happen anytime between 65 and 80 hours post-fertilization. So if the first valve leaflets are 2 cell layers thick, then where do all the valve interstitial cells come from? And here I did an experiment where I stopped the heart at 48 hours post-fertilization. I performed photoconversion using UV light, and I turned some of these cells expressing the green photoconvertible protein K into red K, And afterwards, I return the fish to normal media so that the heart restarts. And then at a later developmental time point, I stop the heart again and I perform confocal imaging. So here you can see this embryo at here, and these cells here have been photoconverted. And at a later development time point, these cells have become the valve interstitial cells. And these cells that have also been photoconverted, they become cells that are facing the blood at the tip of the valve. So putting it all together, it seems that after the cells migrate into the cardiac jelly, you have this reorganization process. And then this layer of cells separates from this layer of cells to give rise to these two-cell thick valve leaflets. The cells that first send protrusions into the cardiac jelly and then form the base of these valve leaflets, they proliferate and they give rise to this first population of valve interstitial cells. And if we compare this to the mouse, rather than this splitting process happening within the myocardial layer, we have a splitting process happening but between two layers of endocardial-derived cells. So now that I've got a clear understanding of how the heart valve forms, I want to find out, do forces play a role in this splitting process? And one of the first things I did was to put a bead into the zebrafish heart to block the blood flow. And you can see here all of these red blood cells, they're going into the ventricle, but they can't go through here. They get stuck, and then so they have no option but to go backwards into the atrium. And when I do this, it completely stops the delamination process. So at 72 hours post fertilization, I expect some of these valves to have already formed valve leaflets. But if I inject a bead at this time point, then there is no further splitting of the valve leaflet. In order to try to have a more subtle way of manipulating forces, I examined the GATA1 mutant. And in these mutants, you have a defect in red blood cell formation. So here in the controls, you can see all these red blood cells going through the heart, but in the mutant, there's no cells going through the heart. And what this does is that it lowers the wall shear stress in the heart, and the cells in this part of the heart, they experience 4 times lower, roughly, maximum wall shear stress. And what I found was that in these GATA1 mutants, at 80 hours post-fertilization, when I expect all of these structures to have transformed into valve leaflets, in the mutants, all of these cells remain stuck together and delamination is significantly delayed. Looking at the cell biology at 80 hours post-fertilization, here in the controls, what happens is that the cells express VE cadherin between all different cells, as well as ZL1, which is a mark of tight junctions. In the mutants, you have a failure to re-express VE cadherin between the cells, and also sometimes you have this loss of the tight junctions as well. And when you have both a loss of the VE cadherin and tight junctions, you have nothing holding the cells together, and you start seeing all these gaps forming between all of these cells. I also performed photoconversion experiments. So here in the GATA1 controls, this layer of cells separates from this layer of cells, and you get these thin valve leaflets. In the mutants, these cells, some of them get stuck inside the cardiac jelly, and then they proliferate, and you end up having these thick hyperplastic valve leaflets. In order to try to find out if what I'm seeing is really due to mechanical forces and not due to some chemical property that's being changed in the GATA1 mutant, I was looking for a way to increase the viscosity of the blood plasma and therefore increase the wall shear stress experienced by the cells. And initially, I was injecting things like starch and dextran into the embryos, but the embryos all died. So I collaborated with Dr. Andrei Klimchenko and his student Valiu, And they had previously developed this nano emulsion that stays in the bloodstream and it's non-toxic. And it's super bright, so they dilute it 1 to 1,000, 1 to 10,000 times, and then use it for angiography. But because I was more interested in the viscosity of this nano emulsion, I just injected it undiluted as much as I can straight into the bloodstream. And what I found was that I managed to get a partial rescue. So here in the GATA1 mutants that I injected with water, the phenotype is pretty much unchanged from the GATA1 mutant that's not been injected with anything. Whereas in the mutants where I injected the nano emulsion, I see that the number of our interstitial cells is significantly reduced, and this is quantified here. So to summarize, in the wild types, you have this layer of cells separating from this layer of cells to give rise to these very simple two-cell thick valve leaflets. And these cells at the base, they proliferate to give rise to valve interstitial cells. In the GATA1 mutant, where you have no red blood cells and lower wall shear stress, the lamination is delayed. And when it does happen, some of these cells get stuck inside the cardiac jelly and they proliferate, and you end up having these thick hyperplastic valve leaflets. So now that I know that mechanical forces are important for this process, I wanted to know what's the mechanotransduction pathway. And for this part of the project, I collaborated very closely with Dr. Hajime Fukui, who now runs his own lab at Tokushima University. And what we are trying to find out is what is happening in the luminal cells, which I'll talk about today, and also what is happening in the abluminal cells, which I won't discuss today. So we know from studies in the mouse that NFAT-C1 is expressed in the luminal cells of the valve, and in the mutant, you have this overabundance of endocardial-derived valve interstitial cells, suggesting that NFAT-C1 suppresses endothelial to mesenchymal transition. We also know from studies in vitro that NFAT activity is dependent on its dephosphorylation by calcineurin, and calcineurin itself is dependent on the presence of intracellular calcium, and it can be inhibited by the drug FK506. So we were wondering if NFAT-C1 signaling could be decreased in the GATA1 mutant. And so I performed photoconversion fate mapping studies in embryos that have been treated with FK506. And what I found was that, just like in the GATA1 mutant, some of these cells get stuck inside the cardiac jelly and they proliferate, and you end up having these thick hypoplastic valve leaflets. And then Hajime created these beautiful movies of that allows you to visualize the calcium signal in the endocardial cells. So here the calcium signal is in green, and the endocardial cells are labeled in magenta. But because it's quite difficult to visualize what's happening in single cells when the cells are not just moving in X, Y, but in and out of the image plane, I created some software that phase matches the cardiac cycle of the beating heart. And hopefully what you can see now is that in these cells labeled with the arrows, the calcium signal actually oscillates. So in Hajime's paper, he quantified these calcium oscillations, and he found out that in the GATA1 mutant, they are significantly decreased, suggesting that they are dependent on mechanical forces. Then Hajime created this transgenic line that allows you to visualize NFAT activity in these cells. And what we found is that in the GATA1 controls, NFAT is activated in all of these cells here. And in the GATA1 mutants, the number of NFAT activated cells is significantly reduced. And here's the quantification. So to summarize, what we think happens is that wall shear stress leads to the influx of calcium, which leads to the activation of calcineurin, leading to the dephosphorylation of NFAT, which then goes to the nucleus and enables transcription. And I don't go through the data today, but we think this leads to downregulation of TWIST1B, which is a major endothelial to mesenchymal transition gene. So its inhibition is necessary for the mesenchymal to endothelial transition, which then allows this process of delamination to occur. And we're quite excited by this because we know that abnormal endothelial to mesenchymal transition is involved in calcific valve disease. And this is the first time that we see cells that are already inside the cardiac jelly becoming re-exposed to the cardiac lumen. So next, now in my lab, we are trying to find out more about these mechanical forces. Previously, I had very much focused on the wall shear stress acting on these luminal cells, but nothing is really known about the forces acting on these abluminal cells, these forces that are embedded in the extracellular matrix. And with work that I'm currently doing with my collaborator, Dr. Yap Chun Hui from Imperial College London, we are trying to map out what these forces are and trying to see if forces could be physically tearing these two tissue layers apart, or if they could be activating mechanotransduction pathways. We're also trying to find out how these cells here communicate with these cells located deeper in the valve tissue. And we suspect there's some sort of paracrine signaling involved, but we don't know what they are. And I've shown this movie before, but what I didn't say was that this was actually a really terrible piece of code. And now that we are trying to look deeper into the calcium signaling dynamics, Raymond from our lab has developed a new version of the software, which has a nice graphic user's interface, so it's very easy to use, and it's much less error prone. And we just submitted this to Bar Protocol, and it has been published. And with that, I'd like to thank everyone from our old lab, especially Julien Vermot, who was my previous supervisor, my past collaborators, all of the IGBMC facilities, and my past funders. And here, I would like to thank Raymond and Zoe, who are students in my lab, Ming Hui, who's an excellent research assistant in my lab, the facilities at Australian Regenerative Medicine Institute and Monash University, my collaborators, and my funders. Thank you very much.
Hi, good afternoon. I think I can't hear you, yeah? So just a second. You're still muted.
How is it now?
Hi, it's perfect. I can hear you. Yeah? So good afternoon, Renee.
Good afternoon.
Thank you very much for the talk. Yeah, so I was pretty much impressed. Yeah, so it's so crazy. Yeah, so I was doing by my PhD something very different, and this is just when I saw this, it was like wow. Yeah, so we're so far that we can image the real heart beating. You know, it's like it's so cool. Yeah. So we have a couple of questions already popping here. Maybe I just before we start, yeah. So I just dropped directly a first question because I was so impressed by this beat in the heart. Yeah. So how is it possible? How do you introduce it? Yeah. So is the fish still alive in this moment, or how does it work?
Yeah. So we surgically inject the bead. So we inject it through the cardinal vein while the heart is still beating. And because there is still blood flow, so the bead basically gets sucked up into the heart on its own. And because the zebrafish is very small, it gets most of its oxygen needs from diffusion in the water. And it can survive even without blood flow for quite a few days. And so it does affect the heart. So the heart eventually collapses, but the fish does stay alive.
Wow, that's crazy. Cool. So I think we would go for the question from the auditorium first. So here we can see directly the first question. So why did you choose glutathione beads specifically? Would other beads work as well?
We were mainly looking for beads that were of the right size, actually. So we didn't need those beads specifically. Although later on in Hajime's work, he actually used magnetic tweezers to move those magnetic beads around in the heart. So in that case, the magnetic properties were quite important.
Okay, I see. So thank you. So let's go for the second question. This calcium channel is the one that forced open by the mechanical forces, right? Is anything known about it?
So we know that the calcium oscillations are dependent on ATP-dependent pyrogenic receptors, so the P2X4 and the P2Y2 receptors. But these, we don't know what's the direct mechanosensitive channel that's involved. It doesn't seem like a lot of the typical channels that you think of, like the piezo or the TRPV4. These channels are all involved in valve formation, but when we inhibited these channels, we still get the calcium oscillations.
Thank you. Then we have one more question. Did you only use them for the viscosity or what the actual purpose? I think the question is about the nano-emulsion, no? Could you remind me what the actual purpose was?
They're very, very bright, these nano-emulsions. So it was actually used for angiography. So it was to visualize the blood vessels.
Okay. Maybe I will.
The fluorescent. Sorry. The fluorescent.
Yeah. Maybe I will follow up on this question because I was curious on the viscosity. So I mean, you introduce it like more or less artificially in this case, but does this changes in the viscosity occur actually in the living organism? Does it affect it?
Yeah. So we changed the viscosity of the blood plasma because the emulsion itself is more viscous. So overall, it changes the blood plasma viscosity about 17%.
Okay. So I have one more question. Yeah. So you showed that in the experiment, I think with the Gata mutant, you showed that the valves got thick in the end. Yeah. So what's the outcome of this having the thick valves? Yeah. So and how translatable it is to the human heart diseases, for example, or like valve associated diseases.
Yeah, so that's one thing that we are really looking forward to go deeper into. Having thick valves is like one of the first signs of calcific valve disease. But what we don't know at the moment is whether this thick valve phenotype that we observe in the larva actually persists in adults. So we're trying to grow up these fish and find out what happens to them.
Okay, but can you see also the grown fish? Yeah, so hard. Yeah, so or is it already too large to observe it? Or
when the fish are adults, they're no longer transparent, so we would not be able to do live imaging. But we would be able to do fixed sections and also to do ultrasound imaging.
okay, very nice. Thank you. We have, I think, one more question. Have you or will you observe development of the coronary arteries?
I haven't actually looked at that, but yeah, that would be interesting to find out in the future.
Okay, thank you very much. Maybe in the last round, very also naive question of Maya. So you also said that there are a lot of research on the mice, yeah. So and I can imagine that such a live images would not also work with the mice, no, because of the probably sizes, no, so of the heart, yeah. So in the end, or
size, yeah, there's a few limitations in the mouse. So the transparency, the fact that the embryo develops inside the mother. So you would have to take the embryo outside of the mother and keep it alive, which is quite challenging.
yeah, okay.
Whereas in fish, the fertilization occurs externally. So the female lays the eggs into the water, and then the male has the sperm into the water, and the eggs fertilize within the water, and you just collect them. You don't have to kill the mother in order to look at the egg, the embryos.
Okay, yeah, I see. Yeah, so the fish is a bit more easy to handle in this case. Yeah, so yeah, so I don't think we have more questions. So I want to thank you again for your great talk. Yeah, so I really enjoyed it. Maybe in the last round you can just outline what's you're gonna be focused in the research wise. Yeah, so what's the road? So where you want to go with your research now?
So want to continue to look at heart valve development and the role of mechanical forces. As I mentioned, that we're quite interested in not just the forces acting on the cells that are facing the blood, but the forces that are acting on the cells deeper inside the valve tissue. We are also trying to find out what happens in valve regeneration, what's the role of mechanical forces in the repair process, and trying to grow up these fish with thick valves into adulthood and see if they recapitulate some of the elements of human disease.
Great. Renee, thank you very much for being with us today. I really enjoyed the talk, and I hope that everyone who watched it right now or gonna watch in the recording gonna enjoy this as well. In the last round here, I wanna also make an announcement of the next masterclass series, which would be on the June 20th, and it will be the talk will be given by Professor David Wood from the Ohio State University, and he's gonna tell us about a quick and simple self-removing affinity tag for highly purified and tagless protein from the single column. Thank you very much for being with us, and I hope to see you again in the next masterclass series. Bye-bye.
Thank you.
Bye-bye.